Hi, my name is Michael. I'm a PhD candidate at the Peter MacCallum Cancer Centre in Australia. And today I'd like to talk to you about the tool we're developing called Isopod, which is a tool that detects differential transcript usage from long-read single-cell data. Now, oftentimes in long-read data sets, we use them because they preserved transcript-level detail. So take, for example, this gene, which has 4 exons connected by introns. Oftentimes what happens is that these genes undergo alternative splicing events. And for example, this gene might produce these lead transcripts, which all have different exon skipping events. These lead transcripts will then go on to produce slightly different protein isoforms, which go on to have slightly different functional effects on the cell. Now, if we just did short-read sequencing on this small data set that we have, we would only be able to get gene expression counts from this data set. However, if we go through long-read analysis, we're able to preserve that transcript level detail and accurately say that we have 1 count of each transcript. Now, what do we do with these? So oftentimes when we have transcript level detail, I think the first thing that we want to do is try to find differential transcript expression. And this is where we just compare the counts between, for example, these 2 cell types. In this case, isoform 2 is the only one that has counts changing between the 2 cell types, so only isoform 2 shows differential transcript expression. However, this isn't necessarily the full picture because every transcript has to be put into context of the other transcripts within the gene that it comes from. And to that extent, we can instead calculate differential transcript usage, which looks at the proportion of the gene counts that each isoform occupies. So in this case, the proportion of both isoform 1 and 2 change between these 2 cell types, and therefore both of them exhibit differential transcript usage. Now, what can you actually use this for? Well, differential transcript usage is oftentimes used to highlight isoform switching events, which is when there's a change in the dominant transcript within a gene between 2 groups. Now, this can happen without gene-level expression changes. So you can have the same amount of counts at the gene level, but those counts can be distributed differently between the isoforms. And these isoform switching events can have functional effects on the cell. And a good example here is in cancer. So we often know that cancer has abnormal alternative splicing events. And that means that in certain types of cancers, there's certain cancer-specific transcripts that can become dominant within some genes. And these have effects on the cell, like pushing it towards a more cancerous state or helping it through the epithelial mesenchymal transition. Now, we also have to consider the single cell aspect of this. In bulk data sets, we often are comparing between 2 conditions. Each condition will often have multiple biological replicates in it. And therefore, when you look at differential transcript usage in bulk data sets, you can actually treat these replicates as replicates. However, single-cell data sets are a little bit different. Oftentimes, you're looking into 1 sample and simply comparing between the cell types in that sample. And in this case, you can't necessarily use the same algorithm to find DTU in bulk than you can in single cell, because you cannot treat every cell as a biological replicate necessarily. And so what's being compared here changes between bulk and single cell data sets. In addition, there are quite a few tools actually for finding differential transcript usage in bulk RNA sequencing. However, there's very few for long-read data, and currently there's only 1 for long-read single-cell data. So to shore up this lack of tools, we developed a tool that we're calling Isopod, which is an R package that we use for long-read single-cell differential transcript usage analysis. What Isopod does is that it uses counts data to find both isoform and gene-level differential transcript usage in a 1 versus all permutation analysis. It's important here to note that this happens after the counts quantifications with tools like Flames or Bamboo. And what you need to provide is just a counts table and a cell assignments table that lets us know which group every cell is in. Isopod consists of 3 main parts: a pre-processing step that removes noise from the dataset, a permutation testing step that I'll get into in the next slide, and a visualization step. For the purpose of this presentation, I will only be talking about the permutation testing step here because that's sort of the meat of what we care about. When you simplify a counts table down, you can really make it look like this for every isoform in a data set, where you have the isoform that you care about and all the other isoforms in the gene. And they have counts that are separated between different numbers of cells, right? In this case, cells 1 and 2 are both belong to cluster 1, and cells 3, 4, 5 belong to some other group of cells. The first thing that Isopod does here is it counts some pseudo-bulk for each cluster. And so we're just adding up the counts within cluster 1 and the other clusters for the isoform that we care about and all the other isoforms in the gene. And what you get is this really nice 2x2 contingency table that you can then pass to a chi-squared and get some p-value for. Now, we're going to remember this p-value, and we're going to go back and do it all over again, except we're going to randomly swap the cells between clusters. Right? Now, when the cells are shuffled between the clusters, the counts are shuffled with them, which means that when you go through the same count-summed pseudo-bulk analysis and get the same 2x2 contingency table, the counts inside are actually quite different. And so you get a different p-value after the chi-squared test. Now, we're going to remember this p-value again, go back and do this 10,000 times. At the very end, we're going to be asking one simple question for each isoform that compares each cluster to all the other clusters. And that question is, how different really was our original p-value? So if our original p-value was very low, and all these other random shufflings produce p-values that were very high, then well, our original p-value is quite significant. Here's an example of that happening. So this is a transcript that we found in one of the test data sets that we have, where the original p-value is extremely low. It's 10 to the negative 17. And this is a histogram of all the p-values that we generated between 0 to 1. Now, if you zoom in on the part near 10 to the negative 17, that black bar is where the original p-value was. You can see that after random shuffling, we're actually getting quite a few p-values that are even smaller than this original p-value. And so even though our original chi-squared p-value was extremely significant, the permutation p-value actually turns out to be not significant. It becomes 0.521 instead. It's important to note here that this can go the other way. You can start with a p-value that's very non-significant, and if all the shuffled p-values that you get afterwards are even more non-significant, that original p-value can then have a permutation p-value that's very low. So the dataset that we tested Isopod on first was Mixology 1. Mixology 1 was a dataset that was published in the Flames paper by Tian et al. In 2021, and it consists of 5 human lung cancer cell lines that were sequenced with both long-read single-cell and short-read single-cell sequencing. Importantly here, it consists of 450 cells between 5 relatively balanced groups of cells here, but it's also very deeply sequenced. We find many isoforms, many genes in these 450 cells, and crucially here, we also find many counts for those isoforms. So we don't run into the same sparsity problem that we may find in normal single-cell data sets. Now, on a macro level, how does Isopod perform? Well, we found that for the Mixology 1 dataset, 562 isoforms were uniquely significant for after the chi-squared, whereas only 35 isoforms were uniquely significant after the permutation. This is interesting because one of our hypotheses for the permutation testing was that it would deflate p-values. And you can sort of see that happening here because permutation is finding less significant isoforms. Now, on the other hand, it's also interesting to note here that we're finding some isoforms that were significant, especially uniquely for the permutation testing. And so we're actually picking up slightly different aspects of the data set here than if you were just doing a chi-squared test. But can we then see these isoform switching events visually? And it turns out that you can. So here is the UMAP of Mixology 1. So every dot is a cell. The larger the dot, the more counts that we have for the gene PQBP1. And we're looking at a transcript within the gene PQBP1. The more blue a dot is, the larger proportion of those gene counts that this transcript occupies. And it's really striking here to see that that bottom right arm, which is the A549 cell line, is extremely blue compared to these other 4 cell lines. And Isopod did, in fact, pick up this transcript as significant for differential transcript usage. This is a very nice, I guess, visual representation of isoform switching that Isopod can find. So we're still working on this. And as a couple of takeaways, I just want to reiterate that we can use long-read sequencing for these transcript level details in single-cell sequencing datasets. We can expose these isoform switching events by using this DTU analysis. There's not that many tools in there. And currently, we're developing an R package that we call Isopod to find these isoforms exhibiting differential transcript usage in long-read single-cell datasets. Lastly, I'd like to acknowledge everyone in the Oshlag lab, especially my supervisor, Alicia Oshlag, and my co-supervisor, Nadia, from the Davidson lab at Walter and Eliza Hall Institute in Melbourne. Thank you.